to study vertical migrations in the ocean, and then transition really to much more about, I think the idea we call frugal science, but another way of describing it would really be in the sense of activism in science. How do you really bring science accessible to society? We're on the brink of a historic election right here. Many of us haven't slept for a long while. And I think it reminds me often again, with or without this election, the importance of engaging in societal context and bringing science to society at large outside our academic avenues. So I think I'll try focusing around more than half the time on that side. Okay, so let's start with the basic science side of the story, which is a very fundamental question we've been thinking about. If you look at life and evolution of life on this planet, you might find this quite intriguing fact that gravity has always been on this planet, 3 billion years or so when life started. Gravity has been present and life evolved in the ocean. One of the questions you could ask, just how does gravity shape life? And I'll describe very briefly, and many of you are from a marine context, so I'm not going to spend too much time on this, but what we're going to try to think a little bit about is the importance of vertical migration and how do we understand and study these really strange ecosystems on the vertical axis in the ocean. So first of all, I get a chance to work with phenomenal students in the lab, and much of the work that I'm describing is done with fantastic graduate students and postdocs. The first work that you'll hear about in the gravity machine is with my graduate student Deepak Krishnamurthy and Hongchuan Lee. One of the things that we find fascinating is people do support some of the science that we do, which is very curiosity driven, which I'm very thankful for, although it's always hard to be able to raise funds for these ideas sometimes. So let's start with to me, my favorite written text ever, both in biology and otherwise, which is Rachel Carson's work on just describing plankton. So I'm just going to read one line out of this, which is, the ocean is a place of paradoxes. It is the home of the great white shark, the largest animal that ever lived, but also the home of small things that your two hands might scoop up, as many of them as there are stars in the Milky Way. And I think what she's talking about is plankton. And then in the very last line, I want to read, drifting endlessly midway between the sea of air above and the depth of the abyss below, there's strange creatures and marine inflorescence that sustains them are called plankton the wanderers. I think it's fair to say even till today that we truly don't understand the biology and the physiology of these strange creatures. We have a sense of the diversity of who lives in the ocean, but what they do and why they do it and how they do it remains a mystery. And I'll explain to you in a second why. Let's take an example of, I know many of you might be cell biologists. You could take an example of, this painting of a rhino that was painted in 1515 in Europe. But nobody in Europe at that time had actually seen a rhino. And so they painted this picture, and some parts of it are correct. But if you were to meet a rhino in a zoo or being chased out in the wild, you would have a very different picture of a rhino. And I think, again, we come back to be thinking about physiology is at the heart of biology. And many times we have failed to appreciate what it is that these organisms actually do, which is, I'm going to focus on that quite a lot today. As a puzzle, I could also just, many of you are aware of the beauty of plankton, but let me pose a puzzle to you. This is a famous painting by Haeckel. Now, I consider this painting as the rhino that you saw before. He saw certain things, he drew them, they are representative, they are the forebearers of what we think is plankton. But was he right? So for example, if I was to just say, did he get the orientation right? These cells have polarity. You can clearly see these crowns of these radiolarians clearly have asymmetry. But with respect to gravity, did he draw them upside down? And I'm going to leave you with that puzzle. I'm not going to tell you the answer until maybe the very end. but just this is a teaser. Keep thinking about it. So let me just dive into what I want to share. I want to share a new microscopy-based tool that we have invented. We call it Gravity Machine. And this is one of the outputs coming out of that machine. What you're watching is Pteria miniata's larva, bat star, a very common starfish that we find on our coasts across Monterey. But what is unusual about this video is you are in the frame of reference of the organism, as if you were floating like a GoPro camera attached to this organism. And this organism is completely free to move, almost infinite distances in the vertical axis. And we can still do subcellular imaging on an organism and even a single cell to a context where these things can migrate, for example, even a kilometer on the vertical axis. And I'm going to tell you the trick soon of how we are able to do this, but also what kind of biology can we discover through a mechanism like this. So this is the Gravity Machine in the team. I already acknowledged Deepak and Hong Chuan. And then on the left is Adam, who's a postdoc, and Delphine, an intern in the lab. So I think I'm going to skip most of this. You all know why ocean is so important. But I think the only thing I want to mention here is how biology connects to the carbon pump. So the carbon pump itself is this biological pump that sequesters carbon, primarily by marine snow and things that die and fecal pellets that go to the bottom of the ocean. It's a very important part of our carbon cycle. But one of the challenges has been is we really don't have the biological viewpoint. These things are not dead particles. These are living ecosystems that sink kilometers. But we truly don't understand the biology, primarily because we have very limited tools that are accessible to us. And in the context of the other aspect of biology, emphasizing the physiology here, is there are hundreds of millions of genes that we have identified in the ocean by just doing next generation sequencing. But we have absolutely no idea, 50% or more of them, we have no idea what they do. So the functional context of this, we are significantly far behind, primarily because we have failed to understand and appreciate the ecological context of life in the ocean, and especially the vertical stratification. And I think finally, this is my final plug to why study plankton is this is my what the hell slide. The aesthetics and the beauty and the shape and form, we truly don't understand why these cells are built the way they are and what are they actually doing. Okay, so let me just jump to the tool itself. So all of you have probably seen this before, the fact that the ocean is vertically stratified. Now it's stratified very strongly. If you think about the thermoclines, the pycnoclines, you can think about density and pressure, nutrients. There is tons and tons of gradients across the surface, across the vertical axis. So for example, and if you think about a biological object like a single cell, the cytoplasmic cell density is roughly 5 to 10% heavier than seawater. So if I was to drop a penny from a boat, you could do this experiment. It would take around 3 hours or so, from my calculation, to reach the bottom of the ocean, say 4 kilometers. But if you were to drop, this is a hypothetical experiment because you'll see the situation gets very complex pretty quickly. If you were to drop a diatom, it would take 2 years, 3 years. If nothing changes, it's the same density. These things sink all the time. But on the other hand, things happen along the way, and we would like to know and understand what happens along the way. And with diatoms, I'm going to actually show you a really remarkable puzzle associated with this, which is, so far we have thought that these are organisms that have no motility, and I'm going to show you that that's actually not true. Okay, so let me just skip most of this because you all are very aware of this. The tool that I'm about to describe, you can think of it as we have had population-oriented mechanisms to study vertical migrations. There are many types of traps that have been developed that you all know that could be put in. But how do you really study biology at a single cell level in the context of the ocean? And I think one of the things that I want to do here is, let let me skip most of this introductory things for you guys. Okay, you've all done microscopy, and one of the challenges of microscopy for the last 200 years, we've been studying life in the XY plane, in a confinement. But I want you to sort of change your perspective now and flip your microscopes. And what I'm about to tell you actually literally allows you to flip your microscopes if you're interested in building this tool. We started thinking about vertical tracking. Could I watch a single cell for its entire lifetime? And how would I actually do that? And it dawned on us that because cells can migrate vertically, we don't even know what types of distances. The traditional approach of making a microscope that would travel along is not feasible unless I have a skyscraper, because the types of migrations we're interested in would begin 100 meters to essentially several kilometers. And how do I watch a single cell in its entire lifetime traversing in these ecosystems that are so dramatically changing without confining these cells? And that dawned upon us as a puzzle when we kept thinking about this for a while. We were studying schistosomiasis that I'll tell you about in a little bit in Madagascar, and I was about to take this flight, and we were building these vertical microscopes to track these parasites for 10 meters. And we were literally packing suitcases with these vertical tubes, trying to think about how could we actually do this. I mean, that experiment failed completely. But out of that idea came about a very simple notion of a treadmill for single cells. What if the two ends of these tubes were actually connected? And so you end up with something like a wheel, and you should think of it, imagine a hula loop wheel. What's very interesting now, we call it the hydrodynamic treadmill, that if the cell is going to swim up, we will spin the wheel down. And if the cell is going to swim down, we will spin the wheel up. So in the frame of reference of the lab, the cell is actually stationary. In its own frame of reference, it's actually swimming. And then we can map the rotational degrees of this wheel to all kinds of parameters, including changes in the ocean. So the changes that we see in the ocean that are in a spatial context, we encode them here temporally. So if the light was changing over time as the cell is climbing up, we just have a lamp on the top that essentially changes light based on the spectra of light that that organism would see. So that was an idea on a piece of paper. We got very excited about it, and we spent essentially 6 months doing math to try to figure out whether something like this is feasible or not. It's actually a trick question because there is a fluid, and hence it can have inertia. And so if you were to spin the wheel the wrong way or too fast or too slow, it would generate essentially a current which would kill the entire device. And the answer is that nature was kind to us. And so this is the actual instrument. We call it the gravity machine. The wheels are made out of glass. We can pressurize them. We can do all kinds of things in the wheel. But I'm more interested in showing you some of the data. There's a lot of math that went behind it that some of you, if you're interested, can read. But there is essentially a theoretical condition that we can calculate to show that if the velocity fluctuations in a given organism are bounded in a certain manner, we can design a wheel that will actually be able to track it. So this is almost, you can think of it as an operating and design parameter. We can not only do the tracking, we can also ensure that the movement is not flipping the cell, because the orientation of the cell is actually very important to us. Okay, so what did we do? We built this tool. We actually started taking them to Marine Station. And now, since I know all of you, I would be very excited about bringing this tool to Dalhousie sometime. The first place we went was Monterey. Then we spent time in Puerto Rico, and we very recently returned from our first cruise in the ocean, actually taking the tool out in open ocean as well. I love this picture because it reminds me of the excitement. Whenever we put any living organism in the machine, we find biology that is just so bizarre to wrap our head around. And now I'm going to just jump in and to show you some of these data sets, because I'm already seeing I'm running out of time, that sort of get me excited about what new discoveries can we make with a tool like this? So first of all, we have mapped around 75 species so far. So if you're interested in this raw data, you can go to the site gravitymachine.org, and you can find all of the data that I'm talking about in raw form. But I'm going to just focus on a few data sets first. Let's start with marine snow. What's fascinating in the context of marine snow is it is a living object. And as a living object, it forms, it grows, there is a whole microbiome associated with it. But we have not had the kinds of tools to really be able to observe this in a context of long distance migration. So we were able to essentially capture fresh marine snow from the ocean at a known height, put this in the machine, and you're watching the very first video of a marine snow being tracked. Literally, the data set here is actually 10 meters. So this is a couple of hours of data set. And in your frame of reference, it's completely static. Very recently, we now have fluorescence microscopy implemented in this, but from this we can immediately calculate the density and the porosity of this marine snow. But most interestingly, we can actually measure how the ecosystem on this is changing as it's going through a virtual environment. So this is almost analogous to Oculus Rift that's associated with the organisms itself. I'm going to switch to embryos and larvae. I think what is fascinating to me in a marine context is this immense diversity of forms and shapes associated with larval forms. I think, the picture to the left reminds you the whole fundamental idea of shape and form is so beautifully connected. And on the very right, we did some work on understanding ciliary bands and why these organisms are shaped this way. And one of the discoveries that we made there is that these bands generate very complex flow patterns. So what you're watching on the right is an actual video of the flow structure. But now that was for a tethered organism. But what happens when you untether and let these larvae go wherever they want to go? So this is the type of data sets that we get. What you're looking here is on the very top is the cellular scale view of that organism. But then if you look in these tracks, you will find something quite surprising. They are all oriented to the gravity axis without any confinement. And what this is a signature of, and I think we've done this around for 70 or so species right now, is the signature of how important gravity is in the ocean. All of these organisms perform a remarkable level of gravitaxis. And this is not just positive, both positive and negative gravitaxis. And remarkably, they do it all in many different ways. So if you were to look at the acorn larva, you can see the tracks are highly complex. That at the smallest length scales, they are exploring, but at the longest length scales, they're still aligned to the gravity vector. While the polychaete larva, for example, which is all the way to the very left, is almost ballistic. I mean, it is shooting up and down, and I'll actually show you some data associated with that in a second. Okay, so let's dive into a few of these data sets. So I'm just gonna switch to maybe one movie. And again, I am skipping all of this because much of this, I mean, the paper just came out, so you can all read as much about this as you want, but I also want to save time for talking about frugal science, which I know many of you want to hear. So we were able to observe for the first time, as far as I know, dial migration at a single organism in a single cell resolution. So this is data for essentially a polychaete worm in day and night circadian context with direct view of that migration. And I'm just going to show you two movies which are my favorite in the context of what really ends up happening in these data sets. So the first is just that same data set that I had shown with Pteria miniata, but I want to show you something quite surprising that will happen. So on the top, you can see the video. On the bottom, you can actually see its climb, and on the very right is its actual trajectory. The red dot represents the organism. This is the trajectory. You know, it's climbing, which is quite a surprise. It knows the gravity vector, but it's doing something just same old for a while. So we'll just let it keep playing. But there is a surprise hidden in this, which is about to come. And one of the things that's fascinating in these systems is this is all data collected with IR. So we have removed every cue that this organism might see in the ocean for this data set first. We have removed light, we have removed pressure, we have removed any thermal gradients, and the organism is primarily using gravity, but it's going to do something quite interesting right around here that we're about to see. So far, so good. The organism is swimming along, and right there is a ciliary reversal event. And what you're going to see, and I'm slowing down the video a little bit, there was that little drop. The organism actually opened its mouth and did a U-turn dive, realigned to gravity, and started climbing again. And for days, this larva will just continue to do this behavior. And this is an innate behavior, and we actually, by mapping the hydrodynamics, we can identify this is actually a feeding behavior. And in a zero information environment, this is an innate behavior that the larva is performing. And again, this is the kind of resolution that then allows us to be thinking about what would be the distribution of these sets of larvae look like. And again, I'm going to have you watch that same behavior just one more time, but now I'll slow that down. You see the same thing. It's about to do a ciliary reversal. There are no boundaries that it's interacting with, and this is the real-time data set. And suddenly it's going to do a ciliary reversal. And what we're interested in is, because the nervous system of these larval forms are fairly simple, in some sense, we're trying to get GCAMP and many neural and calcium markers operating and working to truly understand what circuits actually determine this behavior. Now you can watch that feeding current that I'd shown. There are two vortices, but remember, all of this is done while the organism is completely free to move, because we are electronically tracking the XYZ positions and effectively. And again, this is all done hands off. The moment we lock it into an organism, the organism is locked for a very long while. Okay, so let me jump to some single cell data. And one of the threads that we will focus on is maybe, I don't know if how many of you figured out whether the radiolara did Haeckel was right or wrong. If you want, you can write that on chat. I'm curious, but I'll actually show you the data. This is a single radiolara tract. And actually, it turns out Haeckel was wrong in drawing that orientation, because these organisms have a very strong gravitactic response, but upside down. So the crown, in fact, is actually drawn upside down, which, again, you know, from a biological perspective, we know so little about all of these organisms. But I want to focus on diatoms just for a second to give you a deeper dive into what I meant by completely new biology can be discovered. And then right after this example, I'm going to switch to the other topic. Here's a centric diatom. We started looking at them in Puerto Rico, and you know, we were just expecting to measure their density. But we saw something so remarkable and so jaw-dropping that I still can't sleep sometimes when I think about this data. What you're watching is a single cell that seemingly has been declared to have no motility. It has no organelle like cilia or flagella or something. And it's sinking like a rock, which you would expect, but in a fraction of 100 millisecond, it can freeze itself and suddenly it's buoyant. And this is the fastest density fluctuation that I'm aware of. And then boom, right there, within 30 seconds more, it starts falling again. And it'll do this behavior essentially for hours and hours and hours. We have recorded these data sets for literally, I mean, you think that the movie has stalled, but no, the movie is still ongoing right now, except the organism has figured out to change its density by 5% in a fraction of 100 millisecond. This is, I mean, in my mind, completely new biology. But if you now think about the consequence of trillions and trillions of these organisms, and again, when we do this on the ship, we actually find not only can it stall, they can also go up. The word wanderer that has been associated with plankton, I've been wondering about this for a while now, having seen all of this data, that maybe we got that wrong. Although these are single cell and they seem so almost helpless in the vast ocean. Effectively, if you look at the velocities that are associated for in the ocean that are vertical versus horizontal, of course, the horizontal velocities are very high, but the vertical velocities are quite low. And so you end up with thinking about that these organisms actually truly are tuning. And again, of course, much of what we know about the thin layers that we find of chlorophyll for most of the photosynthetic organisms. And again, the fact that if this organism could not modulate and remain suspended in the euphotic zone, this genome would have disappeared to begin with. So there is an incredible evolutionary pressure for maintaining buoyancy. And we've been teasing apart this mechanism molecularly and working on the ion channels that are associated in diatoms. And I probably don't have any time, so I'm going to skip much of that. I want to show you just one quick video of dinoflagellate, which is known for the toxic algae blooms. This is Akashoa. And if you look carefully, you can actually watch the flagella beating. These movies are kind of a little bit jerky. But one of the fascinating things about this is finally we have a tool at hand to really understand the cellular biology of many of these planktonic forms in their ecological context. We were very excited. So we took this tool to Kilomoana recently before the shutdown. And I'm happy to say that, this is a stable boat, but it was a pretty rough sea at that time. But we were able to operate the entire instrument and work in a context of the ocean. And it was quite a fascinating experience to really be able to build and bring instruments all the way out to sea and make the same kinds of measurements. What was fascinating about this moment was we were just about to hit a storm, and actually the boat had to turn around because the storm became quite violent, actually. It was to a point that, yeah, it was not safe to operate. So in conclusion, I think the thing that I want to say is we're now moving towards implementing all of the ecological context in this wheel, including CTD measurements. So once we get a CTD measurement done at a given place, we can electronically take that and upload that in our instrument. So if you were to put the cell in there, we would be emulating as many parameters as possible. I think currently we are doing pressure, and the picture on the left is sometimes if we're not careful, we can actually explode these wheels. And I think what I want to end this part is just the fact that I am deeply excited about this notion of cellular physiology in the context of environment and behavior, to truly understand what these organisms do and what these organisms are trying to tell us. So on that note, I want to switch and talk about frugal science. The question is, should we take any questions here, or maybe take questions in the very end?
Manu, I think the best thing would be if you kept going and we kept our question to the end. Again, a reminder to everyone, if you have a question that you want to note down, just write it in the chat tool, and then we'll answer everything at the end, I think. Thank you, Manu.
Yeah, I think that might be the best, just also because that way I can take 20 more minutes. I'll take 20 more minutes or so, or 15 more minutes to cover. Now I'm going to go at a very rapid pace. I probably already was talking way too fast. But I do want to share some of these, almost philosophical context with all of you as well. So I think a couple of years ago when I started my lab, maybe around 9 years ago now, one of the things that bothered me was this notion of we are incredibly privileged to have access to science. The idea that we can spend our time exploring our own curiosity is such an immensely powerful, almost soul feeding privilege that we have, but most people don't. And in some sense, if you care about science and science value, not everybody has to become a scientist, but scientific principles determine the way that our lives unfold. And those principles not only have to be known to other people, but possibly even embodied in our day-to-day behavior. And this is a great example of what happened in the pandemic in the United States, for example, with people not understanding why masks can actually save people's lives. Uh, we started thinking about this problem almost 10 years ago, and the goal was, let's not just provide information, let's provide the experience of science. And that, again, becomes challenging because of the cost. So the whole context of frugal science is, if you were to draw any tool that you're using in a cost versus performance curve, how do you reduce the cost by orders of magnitude to a point where they become, I mean, I often think about the tools that we make as the pencils of science, that it should just be so ubiquitous and available without getting a very large hit on the performance. That's the name of the game. Why would you do this? If you think about what's happening on our planet, and again, COVID is a great example for this, we have 1 billion people that just have never had any healthcare access, period, out of 7 billion. This is a picture that I took in Ghana, where this was a health clinic under a bus stop, essentially. Again, most of the field sites in Madagascar that we work in, it takes 14 hours to walk from your village to a health center in an area that's endemic of malaria. So you can imagine what it means to carry your own child, for example, to that distance. And again, we cannot deliver last mile healthcare unless we were to bring these tools to people. You could take and paint the similar picture with education. There are 2 billion kids on this planet, and roughly 1 billion currently live in families that make $2.5 per day or less. That would be the poverty line described in the UNICEF data. Now, this is another picture of a school, and you start realizing that there are no tools of creativity, and hence the challenge that science has is that, there is no actual experience of science that these kids have. And remember, this is literally half of the world's talent. We're talking about not some minor, small minority, and this has nothing to do with developed or developing countries. This has to do with our own backyards, haves and have-nots, and any place I can guarantee you, when you look, you would find communities which just do not have true access to the experience of science. A similar set of a thing could be painted also from the context of what's happening on the planet with the biodiversity loss that's at place that I know many of you think about. Where the hell is all of the pollutants and plastics going in the world? And finally, and last but not the least, just the denial of science overall, which we are facing. I don't have to give any examples anymore. This is just so front and center. So how do you deal with this? These are grand challenges. And in my mind, I think of them as, because they are planetary scale, we need a completely, not just the scientists, which is a very small portion from a human population perspective. We need a much more, firstly, we need a way to measure at planetary scale in the finest granularity. And secondly, we need a very inclusive environment where there could be a true collaboration between professional scientists like you and I and amateur scientists that spend their time in ecosystems and would be willing to truly measure this planet. And this is a hard challenge, but there is a silver lining to this, which is this is not the first time we got forced into this. This has happened before. This is a picture of Sputnik, but I don't know if some of you know the story behind Sputnik and its identification. This was the story of Operation Moonwatch. Fred Whipple, a famous astronomer at that time, essentially launched the world's largest citizen science program, because when the Russians launched Sputnik, we had absolutely no way of tracking whether there is anything in the sky because the sky is so large. Anybody at that time could have claimed that we have launched a satellite that's spying on you. He launched the world's largest citizen science program by shipping telescopes around the world, tens of thousands of telescopes that costed $10. And he trained an army of people. And actually, I love the title of this middle picture. It says, Mrs. Lloyd Eisenhower, housewife volunteer sought Sputnik 2. And actually, for the next 40 years, many Sputniks and satellites that were deployed were first discovered by these community members and then tracked by the military organizations. And it inspired a massive generation of people to really engage in science and look up at the skies. And in my mind, when I think about the community healthcare worker programs and ASHA workers that work around the world tracking diseases, it is no different than a largest citizen science program. It would cost around $1 trillion if we were to pay these individuals. And hence the importance of this infrastructure. And again, if you talk about diseases coming out of the woodworks, this is our frontline defense. This is a picture of Alicia on the right. And the way that I think about our role is, which I'll describe very briefly in the set of tools that we build, is what are the types of tools we can provide in her backpack? You can see she's carrying a backpack, and that's what I'm thinking about. What tools can I provide to frontline workers in ecological measurements, oceanography measurements, and health measurements that common day-to-day individuals could actually use to add to this fight? So we've been doing this for many years now. There's a lot of tools that we have built, all the way from tracking mosquitoes to what Lloyd described about our microscopes and centrifuges. I'll primarily focus on microscopy, but some of this is published, and you can find that on our site as well. And it's important to note that the origin of all the ideas that I'm describing come from us spending time in the field. We spend time in the field, in these conversations lead to ideas. So I'll give you a few examples very rapidly. This is the origin story of Paperfuge, which is the centrifuge that we have developed that costs us 20 cents. This is another version of it. This one is not made out of paper. Essentially, I don't know if you're aware of this toy that you might have played, a button on a string, you put that in, and now I'm essentially spinning a sample, but this is a completely enclosed chamber, as you can see, that's designed for DNA extraction in the field. We built this one for essentially a team that was tracking tigers in India. The story behind this comes from this conversation I had with a healthcare worker in Uganda when they said, you know, we don't even have electricity, so of course none of your centrifuges would work. And on a flight coming back, I was thinking about spinning toys. And okay, I started looking at all possible toys that spin. And of course, we analyzed yo-yos, and then we stumbled upon this remarkable toy, which is called a button on a string. And you've all played with this, but what's fascinating about this actually is the oldest toy in the history of mankind. This is 5,000 years ago. It's been found in all cultures. Interestingly, nobody figured out how it worked. Nobody thought about how it worked. So we took a couple months, and this is how it works. You can write down a single equation that tells us all the parameters. And using this, we were then able to tune this instrument to operate with just purely human power all the way to 125,000 RPM. So that holds the world record for the fastest spinning object with human power. And one of the fascinating things is we can do that for 20 cents and would be equivalent to 30,000 G-forces. So now you might ask, what could I do with these G-forces? First of all, you can detect anemia in 90 seconds. So this is plasma extraction from a capillary. We can pull out a single blood cell that is infected with malaria because the parasite essentially eats up the hemoglobin, and the density gets lower. And using that, you can essentially pull them out in a couple of minutes. And one of the things that I want to mention is sometimes when you make tools, there is a hesitation. But when you bring them out to communities, this is that village in Madagascar that I told you about, which was 15 hours away. And this is the village chief. and the village chief has to give us an approval before we can do a clinical validation study in his village. And there are so many social contexts going on. For example, right behind the village chief is the only woman that's allowed in this room. And culturally, women are not allowed in decision-making places, but she is the community healthcare worker and the teacher of the village at the same time. And so hence she has decision powers. And again, it just reminds us of the importance of empowering all sectors of society to really be able to contribute. When we posted this, lots of open source designs started coming out. There are 3 or 4 diagnostic tests that have been built on this platform. So this is much more of a platform technology that anybody can actually build. We built very recently a COVID test, a home COVID test for a group in Bolivia that is now tracking COVID in the Amazon, which primarily is a LAMP-based assay and uses a similar version of the device, but utilizes these hand flashlights that many of you might have actually used, but essentially replaces the centrifugation step with this handheld centrifuge. I'm going to jump to microscopy briefly. There's a lot that I could talk about for mosquitoes, but let me just spend 2 minutes on microscopy, but then microscopy accessible to sailors. So, of course, to the very left is Foldscope, which is our frugal science, a microscope that we have scaled around the world. Very recently, we released PlanktonScope and Octopi. Those are big brothers of Foldscope. Now, these are automated instrument that costs around an order $100. Octopi is built for malaria diagnosis. And one of the contexts of this is for a price of $100, a completely automated microscopy instrument in which we can do malaria diagnosis. Around in 5 minutes, we can image something like 15 million cells. So what you're just watching is an instrument that is operated on a cell phone battery charger. It has autofocus. It will scan the entire slide. And then one thing that we do very special in this instrument is we can do spectroscopy per cell. And we discovered new sets of dyes that allow us to diagnose and detect malaria parasites with very low resolution imaging. And so this is why we can build this tool. And again, the joy of building these tools is sharing them with communities around the world. So now this is that same instrument now installed in our field site in Kalahandi, and another one installed in Uganda. We're ramping up to essentially build a network, just like Fred Whipple did, of 100 of these robotic instruments that are tuned to malaria at 25 institutes across Africa and Latin America and India. And this is very exciting because this would be the largest data set for malaria collected from patients around the world once we can get this program going. I'm going to show you just very briefly about the context of the ocean and PlanktonScope. One thing we started realizing was that the ocean is a big place and oceanography still is too expensive, and we turned our eye towards sailors. There is roughly around 10 to 15,000 blue water sailing boats crossing the ocean at all times. And we essentially initiated what we call a citizen science program, which is training these sailors first for genomic collection using plankton nets that were provided in the kit. So that first paper is out now. This was from 15 boats. And then very recently, we have now released a tool which we call PlanktonScope. And I'm just going to very briefly show this to you. Much of this is on bioRxiv. It's a stacked microscope that you can build together. It's completely open source. If you go to planktonscope.org, you can find all the instructions for it. But what's beautiful is for a price point of $100, we can literally match the criterias of what you would get out of FlowCam, which would cost $50,000 to $100,000, which is very powerful because now not only scientists, but just an everyday citizen or a sailor could actually collect. So this is a data that we collected in the field in a Bloom. And from a statistics perspective, if you read the paper, you can find all the characteristics and a one-to-one comparison with traditional tools. And what's very exciting is we've been thinking about how to display this data set. So we call these ecograms, which would be these snapshot images. Again, all done completely in an automated manner. And already within the last 2 months, the plankton scope community is starting to grow. There's roughly around 22 units across the world now. And I think I would love for many folks at Dalhousie to be actually engaged as well. So this is the website that many of you can find for this tool. And in the very end, I want to say a word about Foldscope. Many of you might know about Foldscope. It is an origami microscope that you fold together. It costs around $1.75. This is the kind of data from a resolution perspective that you can get out of it. So for a price point of $1.75, you can get a resolution of 1.9 microns, good enough to actually see single bacteria. And one of the things that happens with this, which is really the context of when we released this tool almost 6 or 7 years ago, there was something that was bothering me in some sense, which was, we can make tools, but what does it mean? We can manufacture microscope, but we can never manufacture mentors. And at that time, I made an impulsive decision that we will build 100,000 of these units in the lab and just ship it to anybody who asks for it. And we had no agenda other than just the fact that we wanted to see who would ask for microscopes and what would they do with it. And ironically, at that time, I didn't realize, but what we were doing was the instrument is important, but what was most important was the community. And that led to essentially a waterfall of what has now become the world's largest microscopy community, amateur microscopy community. We have roughly around 1.4 million members now, and many of you hold Foldscopes. And what has been fascinating with this is just the bizarre and the most creative things that people have done with this. There is a centralized repository with literally 20,000 to 50,000 examples of people posting what they discovered. So this is literally the voice of the community. You can look at that. There is roughly around 400 or so papers published with biological findings, sanitation, health applications, all of that driven by the community. For example, this is a picture from a very remote place in a nomadic community with the context of agriculture, where they discovered an application of Foldscope for identifying plant pathogens. Here's a picture of a sailor out in the field who has been sailing around the world using Foldscope, mapping planktonic biodiversity. Here's an example of a health veterinary scientist out in the middle of nowhere for, detecting parasites in elephant poop. This is a community in Iraq. Right after the war ended, Mohammed Abbas, who's one of the Foldscope super users, started a Foldscope club in Iraq in the area that was in a war zone, and many of these students had actually not been to a school for the last 5 years. Here is another picture of one of my favorite Foldscope super users, because after me, he has the most number of Foldscopes. This is an individual who's a teacher in a remote area in Vellore. And in the last 5 years, one single teacher, one individual, has trained, at this current count, 110,000 students and around 12,000 teachers single-handedly, just traveling across the country with a box full of Foldscopes. We keep shipping him microscopes, and he deploys them around the world to an extent that after English, Tamil is the most spoken language on our platform, primarily because of one individual. So I think I find this very empowering, because when I think about it, it's very hard to say how tools and technologies make impact. But what we have to do is trust in the power of humans, give them the tools, and let them explore what they would do with it. This is actually a picture that Mo took, and I'm going to end here. I find this picture just so charming and beautiful in many ways. I mean, anybody who tells you science is not exciting, look at these kids. I mean, they've never experienced science. They're lined up. There's just one single Foldscope. And just, I could feel the chatter and the just, the energy in this room. And of course, this is a picture that I work for because, we've released 1 million foldscopes. In the next 2 years, our goal is to release around 10 million and then 100 million, because remember, there are 2 billion kids. And effectively, foldscope to me is almost like the pencil of microscopy. So unless we can make it accessible globally around the world, we would not have reached our mission. So on that note, I want to end. I really apologize.